analyzing for several heavy metals in vegetables and water simultaneously. The first step is to cut a sample from a vegetable and put it in a vial with nitric acid to digest the sample. Then the digested vegetable residue is removed with a filter syringe and transferred to a new vial. Now an adsorbent is added for the metal ions in the residue to stick to, and a soapy detergent to catch the ions sticking to bits of adsorbent. The mixture is shaken up with ultrasound and spun around in a centrifuge so the detergent can catch the trace metal ions in the vegetable residue. Micelles or clusters form as part of the cloud point extraction process to separate out the ions. A micelle consists of a detergent cage, the clay-based carbon nanotube adsorbent, and the metal ions sticking to the adsorbent. The liquid is poured off and a weak nitric acid is added to release the metal ions from the micelles. The sample is put into ultrasound to shake the ions free of the micelle remains and into a centrifuge to further separate out the metal ions. Then the ions are fed into the spectroscope's filters, so only ions from the vegetable remain. Then the plasma torch in the spectrometer heats up the ions and they start emitting light photons. Each metal has a unique spectral light pattern. Now we know that the vegetable contains metal 1, metal 2, and metal 3, and how much of each. The trace metal amounts can be compared with World Health Organization guidelines or other standards. The research appeared in Food Chemistry in 2020 by Mr. Luthando Nyaba and Professor Philiswa Nomngongo at the Sarchi Chair for Nanotechnology in Water at the University of Johannesburg. The research is titled Determination of Trace Metals in Vegetables and Water Samples using Dispersive Ultrasound-Assisted Cloud Point Dispersive Microsolid Phase Extraction coupled with Inductively Coupled Plasma Optical Emission Spectrometry.
The University of Johannesburg, the future reimagined.